Hi. Today I'm going to talk about some time-resolved experiments that we've been doing on human gamma D crystalline. And we're starting to think of this as a model system for protein UV photostability. So the reason why we're interested in this protein is it makes up the structure of the eye lens. So the eye lens is very interesting because it has to remain transparent throughout our lifetime. The proteins that exist in the central fiber cells of our eye lens are the proteins that you're born with. So the proteins that make up the lens are synthesized in the lens epithelium cells. As these cells age, they differentiate and they lose all of the cellular machinery. And so this is to reduce the scattering of the fiber cells. They then migrate towards the center of the lens, and this is how the lens forms. So the proteins here must remain soluble and folded for the majority of the human lifespan. However, over time, the proteins start to unfold, aggregate due to different types of damage, and this results in a progressive opacification of the lens, which is known as cataract. So cataract is a big problem. It affects about 1% of adults worldwide. But when we think about an aging population, that number jumps. So when people age to over the age of 80, it's about 60% likely that they'll be affected by cataracts. And there's a number of different risk factors. Of course, age and diabetes is also a risk factor, genetics, smoking, and importantly, sun exposure. So currently, the only way to treat cataracts is to perform surgery. And surgeries are extremely successful and require a skilled surgeon to insert an ultrasonic needle into the eye lens to break up the protein. The protein is then aspirated out, and a new plastic intraocular lens is inserted. However, although very successful, the requirement for surgery results in a huge vision inequality throughout the world. So in areas where surgery is quite easily accessible, we see that fewer people struggle with cataract. However, in the central regions around the equator, cataract prevalence is much higher. And if we overlay this with the UV index, we see that the prevalence is higher in regions that have a higher UV index and lower access to surgery. So in my research, what I'm really interested in is the gamma crystallins, which make up the eye lens. There's another structural protein in the eye lens called the beta crystallins, and also a family of heat shock proteins called the alpha crystallins. So the role of the alpha crystallins is to sequester unfolded beta and gamma crystallins to try to prevent them from aggregating. There's also lots of small molecules that diffuse into the fiber cells from the epithelium layer, including a tryptophan metabolite, L-kynurenine. So this is thought to act as a UV filter. Ascorbic acid and glutathione are also present in high concentrations, and these act as reducing agents, trying to keep the redox balance of the fiber cells. What I'm really interested in is this gamma crystalline protein. So gamma crystalline, it's quite small and compact protein, this distinctive Greek key fold. It's present in very, very high concentrations in the lens, so up to about 400 milligrams per milliliter. And it's one of the most thermally stable proteins in the human body. So its melting temperature is about 83 degrees Celsius, and it's implicated in cataract formation. It contains a number of cysteine residues, but despite being very thermally stable, none of these cysteines actually form disulfide bonds in the native protein. We have in the internal N-terminal domain cysteine 18 and cysteine 78, which are quite close together, but never seem to form the oxidized disulfide bond. And in aged proteins that have been taken from cataracts using mass spec studies, a disulfide bond has been seen with a domain shift that actually forms between cysteine 18 and 32 in the damaged protein. We also have a number of surface cysteine residues, C41, C108, and C10. And these are the ones that I'm really interested in the first part of the talk. It also is very, very stable to UV radiation. And this has been ascribed to a number of tryptophan residues. So there's 2 in each domain. And these have been described as a cluster, even though they're not very close together. Tryptophan 42 and 68 are 12.2 angstroms apart, and tryptophan 131 and 156 are 11.5 angstroms apart. But due to slightly different electrostatic environments, their absorption spectra slightly shifted, and it's thought that this promotes a FRET energy transfer, allowing dissipation of the absorbed UV energy throughout the protein and reducing UV-induced damage to these tryptophan residues. However, it's been shown that the conversion of tryptophan to kynurenine through UV damage is present in damaged human gamma D crystalline. So really, when we set out to do our time-resolved studies, what we wanted to see is if we could induce this damage and see what effect it had on the early stages of protein damage prior to unfolding and aggregation. So being interested in X-ray crystallography, we've shifted. Initially, X-ray crystallography was very powerful for helping us to understand protein function by looking at structure. But when John Kendrew first suggested that he wanted to crystallize proteins and understand their structure, he was criticized because crystals don't wriggle, and if it doesn't wriggle, it's not biology. But luckily for us, the proteins inside crystals can wriggle, and we can see how dynamic changes link structure and function. So to do this, we use time-resolved crystallography, which has already been introduced in the previous talks. And we like to do these experiments over a range of timescales. So if we're looking at the very fast photochemical changes, we need to use X-FEL radiation to get down to the nano, pico, and femtosecond time regimes. However, we can also use synchrotron radiation. So Lowy experiments were really the first time-resolved experiments that were performed in earnest, and Lowy radiation can get down to picosecond time resolutions. And we can also still use synchrotron monochromatic beamlines if we're interested in the millisecond to microsecond time regime. So in our experiments, we're really interested in understanding how the very fast photochemical changes can then result in structural changes throughout the entire protein at slower time scales. So using our typical pump probe technique, we trigger our reaction with a laser pulse, and after a fixed time delay, we probe how the structure has changed over time. And this has been covered previously, but there's lots of challenges in these experiments. So we need to do our data collection at room temperature if we're going to see dynamics and structural changes. So that means we can have problems with radiation damage. Initiating reactions is often difficult, and problems with signal to noise ratio. And the time resolution really is determined by how long it takes to initiate our reaction, the intensity of our source, and the type of detector that we're using. So the synchrotrons, as I mentioned, we tend to look at the slower changes and then use the X-FEL to look at the faster changes. In the experiments that I'm going to talk about today, we use fixed target data collection. So that means we have chips. These contain 8 by 8, what we call city blocks. And within each city block, we have 20 by 20 wells in which we can pipette crystal solution. And this is quite a convenient way to do our experiments. We align on each crystal position, shoot first with the laser, and then a fixed time delay probe with our X-ray beam. So here you can see the setup that we use on Beamline I-24 at Diamond Light Source, but we can take this setup and use it at Sackler XFEL in Japan. And there's also a very similar setup at the Swiss FEL, which Petra mentioned. So we need tens of thousands of microcrystals in order to be able to solve our structure. And luckily for us, but unfortunately for the patient, a mutant of human gamma D crystalline was isolated from the eye lens of a 5-year-old boy. And here you can see the lens. And from the lens, crystals of human gamma D crystalline were extracted. So this is a crystallization mutant that causes crystal cataracts. And the mutation was actually determined by X-ray crystallography. The crystals were taken directly out of the eye lens, placed into the X-ray beam, and the structure was solved. And then that mutation was then confirmed by genetic analysis. So we use this mutation in order to be able to grow crystals of our protein very quickly and in very minimal conditions. So we simply have 100 millimolar phosphate buffer, pH 7.8, and 20 millimolars of reducing agent DTT. And our crystals spontaneously form around 25 to 50 microns, depending on the temperature we use. And then we load them into our chips under red light prior to our experiment. So the first step in any time-resolved experiment is to get the dark state. And so we weren't expecting to see this in our dark state, but what we observed when doing the serial crystal experiment with lower doses than had previously been used to solve the structure was a disulfide adduct that had formed on 2 of our surface cysteine residues. So we confirmed that the disulfide adduct was actually the DTT, had bound to cysteine 41 and cysteine 108 in our structure. We then so we wanted to know if this caused any significant structural changes in the protein and if the disulfide adduct had actually affected the fold. It's quite difficult by analysing the overlays of our 2 structures whether there were significant structural changes. So to do this, we actually used a program that was developed by Helen Ginn to use torsion angles throughout the structure of the protein to perform a principal component analysis. We then took a linear regression of the torsion angles deviated associated with the principal component, and what we saw was an expansion of the N-terminal domain and a contraction of the C-terminal domain in our protein. And using that torsion angle description of our protein, we were able to calculate structural paths that show realistically how the protein modulates between the adduct-free and the adduct-bound structure. So here we can see a sort of breathing motion with large structural changes or significant torsion angle changes highlighted in the lighter red and yellow colours as we change from our fresh to adduct-bound structure. We then took our crystals to Diamond Light Source and we exposed the entire chip to UV radiation. So we simply used a LED at 285 nanometers. And what we could see is removal of the disulfide adduct upon exposure to UV radiation, and a reversal in the changes in our structure. So we saw then a contraction of the N-terminal domain, an expansion of the C-terminal domain. And again, by calculating paths, we could see the opposite effect of the breathing motion in our protein as it relaxes back down to its native conformation. But what we really wanted to do was understand whether these structural changes and the removal of this adduct returned our protein back down to its ground state. So to do this, we performed a batch analysis of our data. So when we do serial crystallography, we inherently oversample. So we need to take tens of thousands of data sets of diffraction images in order to be able to solve the structure with reliable metrics. However, we can take advantage of this oversampling in order to calculate our structures. And so by taking a subset of data from each chip, we split our data sets into 5 equal bins. We then performed the principal component analysis, and we could see 3 distinct clusters when we compare our fresh crystals, our aged crystals, and our UV exposed crystals. So there's an obvious change in the overall structure, not just in the position where the adduct binds between these 3 different samples. So what we saw over time was that our crystals aged, a disulfide adduct formed, and then after 5 milliseconds of UV exposure at 285 nanometers, the adduct was removed. We wanted to know what mechanism was taking place here. So did we have direct photolysis of a disulfide bond, or was another process taking place, perhaps involving the neighboring tryptophan from each of our disulfide bonds? So we looked more into this, and what we found was that actually the carbon-sulfur carbon dihedral or torsion angle greatly affects not only the maximum wavelength of absorption of disulfide bonds, but also the extinction coefficient. So we tend to think of disulfide bonds absorbing around 250 nanometers, but as the disulfide bond becomes more strained, the maximum absorption increases in wavelength, and the extinction coefficient drops dramatically. So looking at our disulfide bonds of our 2 adducts, we found at position C41, the disulfide bond was less strained. It had a lambda max of around 260 nanometers, an extinction coefficient at 260 nanometers of about 684. It's reduced when we go down to 285 nanometers. However, the disulfide bond that's closest, at position C108, is actually much more strained. The lambda max is 342 nanometers, and the extinction coefficient at its lambda max is 195. And so what we're interested in understanding is actually, is there a relationship between the neighboring tryptophans to each one of these disulfide bonds, and is there actually a quenching effect whereby we get some kind of transfer from the tryptophan to the disulfide that results in the removal of the adduct? And so the distance between our tryptophan 42 and our C41 disulfide bond is 6.7 angstroms, and the distance between the cysteine 108 adduct and the tryptophan 131 residue is 8.3 angstroms. So this is within the distance that electron transfer might happen. And we know that tryptophan is much more absorbing at 285 nanometers. So one of the main points here is, although we're looking at adducts with a non-native reducing agent, DTT, we see the same formation of an adduct with the native reducing agent glutathione. So the concentration of glutathione in the eye lens is about 10 millimolar, and in previous research using mass spectrometry, S-glutathione-ylated proteins had been observed in aged lenses. It took us a while to get the bound structure of glutathione, but we finally managed it, and using extrapolated difference maps, the program Extrapolate, which Elka will be talking about later, we found an occupancy estimate of about 0.4 of our glutathione at cysteine 108. So the glutathione is much more dynamic than DTT. There's more conformational sampling that can occur, so it's quite difficult to capture this structure. So most of our research, we are focusing on using the DTT as an analog. So the binding of the reducing agents really occurs due to the formation of reactive oxygen species over time and oxidation of our cysteine. So in our protein, you'll also see that at position 110, we see the sulfenic acid oxidation product of our cysteine. This is quite interesting, as sulfenic acid is supposed to be very unstable and is either further oxidized or reduced by GSH back down to cysteine. So over time, what tends to happen is the concentration of glutathione is reduced and we get an increase in the oxidized dimer GSSG. And this GSSG will then bind to sulfenic acid for long periods of time if the concentration of GSH to reverse that binding is lower. So in order to try to understand whether it's direct photolysis of our disulfide bond, or if in fact we're seeing an electron transfer, we need to start thinking about doing different spectroscopic measurements, and thinking about different mutations. So far what we've looked at is the wild type structure and the W131A mutant. And what we see in the wild type structure is that the addition of these reducing agents combined with aging results in a decrease in fluorescence of the protein. So this decrease in the fluorescence indicates that binding of the reducing agents results in some kind of quenching mechanism. So this suggests that maybe the tryptophan is being excited and the disulfide bond can then quench that through the removal of that adduct. And we've also seen that upon binding of our reducing agents, we don't get any interaction with the thiol probe Elman's reagent, or DTMB. So DTMB binds specifically to the SH group in thiols. And however, after UV exposure, the sulfur on our cysteines is then free to bind to the DTMB, resulting in an increase of absorbance at 412 nanometers. So we then wanted to see, because what we saw was a ratio of our DTMB binding to protein of 3:1. We know there's 3 surface cysteines that are solvent accessible. However, one of them we didn't think was a thiol group. We thought it was a sulfenic acid group, so we wanted to see what effect that was having after UV exposure of our proteins. And actually what we found was binding of the DTMB occurred at W42 and C41, sorry, and C108. However, we saw no evidence of binding at our C110 position where we have the sulfenic acid. What we did see, due to an expansion of that N-terminal domain, was exposure of the previously buried cysteine 18 to the solvent through the flipping of this tyrosine 16 out of the way, allowing binding of our DTMB. So we're quite interested now in this cysteine 18, as it's also implicated in the domain switch and the formation of a disulfide bond. We collected our first XFEL structures about in 2023. At that point, we were supposed to do some UV initiation, but the pump laser wasn't working. So we collected lots of dark structures. And so we collected our dark glutathione structure. So we had a time point for subtracting for our difference maps. And what we found is when we only have the 25 femtosecond exposures per crystal, we were able to obtain what we're thinking of as our sort of damage-free structure. And at C18 and C78, we have some interesting density. So we don't have a direct disulfide bond between our two cysteine residues, but there's sort of a bridging between them. And it looks to be modulated by two extra atoms. So our current working hypothesis here is that actually we can form sulfenic acid at these two positions, but only when glutathione is bound to the structure. In our structures that contain no glutathione, we don't see this. And we think that's due to that expansion of the N-terminal domain and the movement of the tyrosine. So we're currently thinking of these being sulfenic acid and a potential hydrogen bond forming between the oxygen group here and the OH group here. Although we need to confirm this, but we're not entirely sure how to do that because sulfenic acid is very unstable in mass spec. We need to do some different spectroscopic measurements here. So the next experiments we want to do at Swissfel will use the pump laser. So in order to use the UV radiation, we've had to build a secondary line. The optics in the normal pump laser line that follow the path of the X-rays aren't UV active, so we have to take our UV laser in through a sort of on-axis excitation path, but we should be doing these experiments in June. So we have our R37 crystals that we do the experiment on, and we also were able to collect a structure of our W131A mutant, which you can see here in purple. It looks as though overall the protein structure doesn't change, just the arrangement within the crystal shifts a little bit. So hopefully then we'll be able to see if we get the removal of the disulfide adduct at the C41, and if we get it at the C108 in our W131A mutant, we're going to assume then that potentially W131 is not actually involved in the UV removal of the disulfide adduct. Really, if we want to be able to understand the mechanism properly, I think one of the best ways to do that is for us to do X-ray absorption spectroscopy. So we've teamed up with Niels Huser at the University of Hamburg, and the first experiments that we've done were performed on cysteine. So that's just the simple dimer of cysteine. And we could see direct photolysis of cysteine at 267 nanometers. So here in our first spectrum, we have the static spectrum. We can see our disulfide bond peak here. And then after exposure at 267 nanometers at 300 femtoseconds, we see the formation of our thiol radical very quickly. And then after 10 picoseconds, we seem to see rebinding, but then breakage of the CS bond and a per-thiol radical formed. So to do these experiments, we've used the PAL XFEL, Swissfel, and EU exfil. These photos are taken from the experimental setup used at EU exfil. It's pretty much the same setup. We have a jet of protein and our laser excites through the jet, and then after a particular time delay is followed by our exfil pulses. So for cystine, the jet worked really well. These experiments were quite easy to perform. However, for the proteins, it's much more difficult. So to get a reasonable signal to noise ratio at the sulfur edge, we need about a 5 millimolar concentration of protein, and the sample consumption's quite high. So we need 50 milliliters of 5 millimolar concentration of protein. So for lysozyme, the experiment that we've done so far, that's quite straightforward. So we can buy that quite easily. However, for us to be able to produce the crystalline protein in those concentrations, so far we haven't managed it yet in time for an experiment. But lysozyme also has disulfide bonds close to tryptophan residues, and we've tried excitation at a number of different wavelengths, but when we excite at 290 nanometers, after 300 femtoseconds, we see the formation of this thiol radical, as we saw in the cystine. However, after 3 picoseconds, we see an increase in this peak here. So this was absent in our cystine experiments. And we think that this peak can be attributed to a thiol anion. So this thiol anion is indicative that an electron transfer has taken place. We can't say exactly where from, but we can assume some of the disulfide bonds within lysozyme are close enough to tryptophans that would seem to allow an electron transfer mechanism to take place. So currently we're still waiting to do the full-time resolved experiment with the laser at Swissfeld for our structures, and we are attempting some methods development to allow us to use the crystalline protein crystals to enhance the X-ray absorption signal. So if we can do that experiment using crystals rather than solution, it should be much more straightforward. So I'd like to thank all of my group that have been involved in these experiments. So Lisa Pulnova is my PhD student who works at Charles University in Prague and is a beamline scientist at the Extreme Light Infrastructure. Jake Hill, who's at the University of Bradford, and this is my MChem student from last year, Audrey Rose Skinner, who did some of the experiments with us at Swissfeld. I'd like to thank Helen Ginn for the software and the support that she's provided in using that for doing our torsion angles analysis, and Niels Huse and Jessica Harrich for their help with the X-ray absorption spectroscopy experiments. And thank you all for listening.